Hi, I'm Sadashiva Pai from Science Mission. Today we have Dr. Schafer, Associate Professor in Department of Biological Sciences at University of Notre Dame, Indiana, is with us. Dr. Schafer has won several awards to his credit over last several years. I'm going to briefly mention some of them. In 2014 to 2018, he won a Research Scholar Award from American Cancer Society. In 2014 to 2017, he was the Career Catalyst Award from Susan G. Coleman Foundation. 2011 to 2013, he was the V Scholar Award. He won the V Scholar Award from V Foundation for Cancer Research. 2009 to present is the Coleman Foundation Collegiate Chair of Cancer Biology, University of Notre Dame. 2007 to 2009, Ruth Christian National Research Service Award Fellowship from National Cancer Institute. So he's also in the advisory board of various publications, and he has also won several grants and other awards to his credit. Welcome, Dr. Schafer, to Science Hangout.
Thank you for having me. It's great to be here.
Can you please talk about yourself? Tell us about yourself, your background, your interest, everything.
Yeah, sure. I'm originally from Springfield, Illinois. I did my undergraduate work here at Notre Dame. And then I was a graduate student at Duke with Sally Kornbluth, who's renowned for her sort of cell cycle and cell death studies. And then I was a postdoc at Harvard Med with Joan Brugge, and where I studied metabolism and matrix attachment. And Joan is really well known for her studies on breast cancer biology. And then I've been back here. I've been back here at Notre Dame, which again was my undergraduate alma mater. I've been back here since 2009.
Good. Just talk us about a little bit about your mentors, role models in your career.
Yeah, I've been really privileged to work with to have some really phenomenal mentors. And I'll start just starting with Sally Kornbluth, my doctoral advisor. She really taught me how to be a scientist and really the basics of how to design experiments, how to interpret data. And she created a lab environment that was really dynamic, really exciting, really great to be a part of. And she, there's no question that she has influenced me as a scientist tremendously. And my postdoc advisor, Dr. Joan Brugge, she really got me more into cancer research, cancer biology. She is I can't say enough about her as a mentor. A phenomenal mentor who cares tremendously for all of her trainees. It was a privilege to be part of her lab, and she's just again, the dynamic that she creates in her lab is one of creativity, one of asking questions, one where people feel free to exchange ideas, and I was just thrilled to be part of it. I've shaped as a scientist.
I know you are a busy scientist. I know you have family and all those with kids and all. So tell me, please talk about your hobbies and other things, what you do other than the research.
So yeah, I have a big family. I actually have 5 kids. They range in age from 12 down to a newborn who's only 4 months old. So really, those are my hobbies. I spend pretty much all of my time with that I'm not here. I'm with my family, with my wife, kids, and they're amazing. They're tons of fun, and we really we always have a great time. And to be honest, they really keep me balanced. I think everybody who works in science understands that it's a challenging job. It's also an amazing job, but it's really amazing. It's I think really great to come home and be able to hang out with those guys. And I think they really balance me and help me to be a better scientist by keeping always reminding me to sort of maintain perspective on day-to-day importance. So that's really they are my hobbies right now. No question.
Great. Nice to know that.
Yeah.
And let's talk about your lab and research
Yeah.
projects.
So one of the things that I really like about a place like Notre Dame is it's a phenomenal graduate student community. I've had really amazing graduate students the entire time. My lab has been open about 9 years now, and
Mm-hmm.
I've just been really fortunate to have been able to recruit phenomenal students. I've had 4 PhDs, who are 5 PhD students who graduated. I've got 4 PhD students in the lab right now. I've got a technician, and I've also got several undergraduate students. So I really wanted to be in a place where I could be involved in graduate education. Um, I think this is just a phenomenal place to do it, and I've had phenomenal people in my lab from the start. I'm really lucky that that's been the case. So we have, I think, a really dynamic group. A lot of curiosity, a lot of people working really hard, and so yeah, I've really been sort of I've intentionally had a very grad student focused lab since we started in 2009.
And you mentioned that you got into cancer research from your postdoctoral work and all. So
Yeah,
talk more about it, and maybe you eventually can get into your presentation.
sure, of course. Yeah, I mean I was certainly interested in cancer when I was a graduate, and certainly I was personally affected, as I think most people unfortunately are. But I think it was sort of a scientific curiosity that got me into it. I was really interested in cell death and cell death pathways when I started as a graduate. I became more interested in the applications of how cancer cells will resist or turn off cell death pathways. I got interested in that early. And then when I started my postdoctoral work, I really started to look at that in the context of tumor progression and also the contributions of metabolism cell death pathways. So I certainly, it's, again, there is certainly, I think for a lot of people or for everybody, really fun personal motivation. But I honestly might maybe trace my path in curiosity in biology, and the questions I found myself drawn towards were really focused on understanding cancer.
So please go ahead and present your work.
Perfect. So I thought what I'd do is talk a little bit about my lab and more broadly, and I thought
Okay.
I'd talk a little bit about some of the recent data that we've got.
Uh-huh.
All right. So let me just bring up my slides. All right. Those up there, okay?
Yeah.
Okay. So my lab is really interested in understanding how cancer cells live and die as a consequence of their status of being attached to the extracellular matrix. We're really interested in metabolism and how metabolism contributes to those decisions about whether a cell lives or dies. Um, I like to use this as sort of a framework to understand where we are, where my lab is in the context of studying cancer. This is from The Hallmarks of Cancer by Doug Hanahan and Bob Weinberg. Um, it's you can see there's all of these different hallmarks of cancer. People are studying a variety of these different hallmarks. Where we are is really in we're sort of the intersection of three of these hallmarks. The first being understanding invasion and metastasis. Second being invasion of cell death, which again I've been studying cell death since I was a graduate student. And we're fundamentally interested in understanding the regulation of cellular energetics. So we're really interested in sort of the intersection of those three of the whole. Now, again, I've sort of talked about this idea of attachment to matrix. So we're interested in cancer cells and how they survive or die depending on their status of whether they are attached or not attached to extracellular matrix. And this is just a schematic that I use often. But extracellular matrix, this is a meshwork of proteins that are secreted by a variety of different cell types. You're thinking about things like laminins and collagens, and most cells will utilize integrin receptors to attach to the extracellular matrix. Now, if they lose that attachment to the extracellular matrix or to the ECM, they will undergo a cell death process known as anoikis. And anoikis is a Greek word for homelessness. So the idea is these cells are undergoing programmed cell death as a consequence of they're undergoing programmed cell death as a consequence of losing that attachment or departing their home. So importantly, anoikis is a caste-based dependent, so it's classical programmed cell death, classical apoptosis as a consequence of detachment to ECM. And as you might predict, if you think about cancer cells and the fact that they're gonna encounter a variety of different microenvironments during tumor progression, cancer cells typically will acquire some type of resistance to anoikis. Now, one of the models that we use was initially developed by my postdoctoral advisor, Joan Brugge, and sort of inspired by the work of Mina Bissell, where you can use MCF10A cells, and these are immortalized but non-tumorigenic mammary epithelial cells. So that if you inject them into an animal, you will not get a tumor, but you can grow them in a fashion that allows you to study an architecture that's relevant in the context of the mammary gland. So if you grow MCF10A cells in Matrigel, which is a commercially available sort of matrix preparation, over time you'll get these spherical structures here, and over time those spherical structures will hollow out, and you can you have to sort of imagine that you're looking at a cross section here, like an optical section, where basically those cells in the middle will die out and you get a hollow lumen. And this is typically what you would see in the context of the mammary gland. Notice that you see this red line here. That red line represents the basement membrane, which is made up of extracellular matrix proteins. And only those cells that are touching the red line or are attached to the matrix are surviving. So this has become a really good model to study matrix detachment, like the cells in the center would be, induced cell death, and in a physiologically relevant context. Now, what's sort of interesting is if you can manipulate these cells, you can put in something like BCL2 to block apoptosis, and that blocks classical anoikis. And what you'll get is basically over time, you'll see a difference where the MCF10A cells will form hollow lumen, and you'll get a filled lumen when you express BCL2. And so that's sort of expected. BCL2 has shut down the anoikis pathway. But what's really interesting and got us thinking about some of the work we're doing now is that if you wait longer, those cells, the ones where you have blocked, if you've blocked apoptosis or you've blocked anoikis, those cells will die as well. And what that tells you is that it's not enough to simply block anoikis or block apoptosis in order to get cells that survive in the lumen, that there has to be some sort of alternative cell death pathway that's clearing those cells from the luminal space. Now, interestingly, if you put in an oncogene like ERBB2, so ERBB2 overexpressed in 25 to 30% of all women who have breast cancer, widely studied, the target of the drug Herceptin. If you do this experiment where you put in ERBB2 into the MCF10As and you get these multi-acinar structures. So again, you get very different looking structures. But pay attention here to the individual structures where you can see now, instead of having a hollow lumen, you've got cells that are now populating the space of each of these individual mammary acini. And so what that tells us, or the way that we've interpreted this, is that ERBB2 has through some downstream signaling, has the capacity to give matrix detached cells long-term survival. So it must be able to block anoikis, and at the same time, it must be able to block this alternative cell death process. Now, if you think about well, what could potentially be happening? It turns out that matrix detachment, again, what this means is cells that lose that integrin-mediated attachment to matrix proteins. When cells lose matrix attachment, there's actually a variety of different cellular changes that can happen. So I talked about anoikis. Again, anoikis is classical programmed cell death, classical apoptosis. In some contexts, it can be regulated by a downregulation of the EGF receptor and an upregulation of the proapoptotic protein BIM. But in addition, there's a variety of other things that can happen as well. So a colleague of mine, Mike Overholzer, who's now a PI at Sloan Kettering, discovered a process called mitosis, where matrix detached cells can actually invade into their neighbors and then can be killed when inside of another cell in the lysosome. We've seen evidence in a variety of different papers that there can be changes in the differentiation status, where matrix detachment can induce sort of a squamous-like differentiation. And most importantly, for what I'm going to tell you about today, is that you can see evidence of something called autophagy, which is typically thought to be sort of a starvation response. And this is largely work that's been done by Jay Debnath at UCSF. So if we think about the fact that matrix detachment induces autophagy, then this brings us to this question that maybe these cells, matrix detachment is also a signal for some type of changes in nutrients and how cells utilize nutrients. And what I mean by that is, when we do these experiments and you see autophagy happening in matrix detached cells, we're giving these cells a full complement of, of nutrients. We're giving them glucose, we're giving them glutamine, et cetera, and yet they're undergoing autophagy. So this suggests that maybe these matrix detached cells have some sort of a deficiency in their capability to actually process nutrients. And this is going back to some work that I did in Jones' lab back in 2009. But one of the things we found is that matrix detachment itself is a signal to fundamentally rewire cellular metabolism. So if you follow me through this here on the left, if you look at normal cells, and what I mean by normal here, I mean non-cancerous cells, there is a deficiency in glucose uptake. As a consequence of this deficiency in glucose uptake, you see a limitation in flux through the pentose phosphate pathway. The pentose phosphate pathway is critical for the production of NADPH, which is important for regulating oxidative stress. So as you lower NADPH, you get an accumulation of reactive oxygen species, which can impact energy generation and itself cause cell death. Now, in contrast, if you look at ERBB2 transformed cells, so these are cells engineered to overexpress ERBB2, you can restore glucose uptake, restore flux through the pentose phosphate pathway shown down here, elevate your NADPH production, and thereby reduce or start to mitigate those high levels of reactive oxygen species, which can function in a pro-survival effect to promote the survival of these cells. So again, what I want you to take away from this is that basically matrix detachment itself, in addition to signaling to cause anoikis, which is caspase-dependent cell death, it's also a signal to fundamentally rewire metabolism. And as a consequence of how that metabolism is rewired, you can ultimately get changes in whether those cells live or those cells die. And really what that leads us to is this idea that cancer cells have to do really two things. I'm sort of oversimplifying this, but just conceptually, this is a good way, I think, to think about it, is that cancer cells, if they encounter matrix detached conditions, they have to both block anoikis and they have to sort of rectify their metabolic deficiencies or rewire their metabolism in a way that promotes the survival. And what we know is that that sort of metabolic rewiring and the induction of anoikis, that those are independent of one another. These are not pathways that depend on each other. So we have two separate things going on here. We've got cancer cells needing to block caspase-dependent cell death and anoikis, and you need to rectify or rewire your metabolism. If you don't do both of these, it's not enough to give you long-term survival. So we're really interested in these questions. So we're interested in how it is that cancer cells regulate anoikis and how it is that cancer cells will rewire their metabolism. That's a fundamental question that my lab is interested in. So what I thought I'd do is go through some of our recent data today And it's really focused on two key questions. How is it that matrix-detached cancer cells rewire their metabolism? And two, what is the mechanism by which these cells ultimately undergo cell death if it's independent of anoikis? Okay, so one of the things that we thought about is if the cells are not dying by apoptosis, then perhaps they're dying by some sort of alternative pathway. Potentially, they're dying a necrotic death. And it's become clear over the years that necrosis, much like apoptosis and anoikis, match with the genetic program. And so this work really originated with Zheng Yuan probably almost over 10 years ago now, where the conceptual advance was that programmed cell death is not just apoptosis, but rather programmed cell death can include a necrotic death, which was typically thought to be sort of accidental or incidental. The term that's been used uh, you know, and I'm going to show you a story up by graduate in my lab. And Mark really was the main driver of this project and the data that I'm going to show you here today. So basically, what Mark did initially was asked sort of a very straightforward question. Does matrix attachment serve as a signal to activate RIP kinase 1? And we did this a couple different ways. I want to focus on the top left here. Just there, we did this so that they would not die by classic anoikis, and we could focus instead on that non-apoptotic or non-canonical. And we grow them in either attachment conditions. And to do attachment conditions, what we do is we grow them on a plates that are coated with polychema, which basically forces these cells to grow convention. And what you see is here, if you look at RIP kinase 1, there's a significant induction in RIP kinase 1 when you grow cells in detachment. And that RIP kinase 1 is active, as indicated by the phosphorylation of RIP kinase 1 at series 1. So it looks like detachment itself is a signal to lead to the upregulation of RIP1 and the activation of RIP1 as well. And then we moved on to the next question being, well, does that upregulation matter? Does that RIP kinase signaling actually impact the viability of cells from attachment? And if you look down here on the bottom left, this is a cell viability assay where we're measuring the reducing potential of mitochondria, and we're using a small molecule inhibitor of RIP1 called NEC1, necrostatin-1, blocks its kinase activity. And you see a significant increase in the viability of cells when you treat with NEC1. And that, importantly, that happens only when cells are detached. If you do this experiment, and we sort of normalize the viability in this case, we'll measure the detached over attached viability. But if you do this in attached cells, treatment with NEC1 literally does not matter. It doesn't impact the ability of those cells to live or die. So this is something very specific that happens only in matrix detached cells. If we do this in that three-dimensional culture model that I introduced to you before, where we're looking at the ability of cells to populate the luminal space of mammary acini, you can see here we've done this experiment where we've got our MCF10A cells treated with the DMSO control or treated with NEC1 to block RIP1 kinase activity. We stored our three-dimensional structures into these four different categories. And you can see controls. They're all basically scoring as clear or mostly clear. You treat with NEC1, And now they're scoring, many of them are scoring as filled or mostly filled. There's substantially more with at least some cells populating the luminal phase. You do the same thing with cells expressing BCL2. And basically, this is a longer period of time, but over that time, again, you'll start to see most of those structures eventually clear. That's shown here. And then when you treat with NEC1, you now shift to structures that are filled or mostly filled. So basically, both in our attached versus detached experiments, as well as our three-dimensional culture assays, you see fundamental changes in the ability of these cells to survive in the lumen of these three-dimensional structures. Okay, so I showed you this in MCF10A cells. Again, I told you before, these are cells that are basically, they're immortalized, but they're non-tumor-agenic. They've not. So we wanted to do these similar experiments in cancer cell lines, and we actually did this in a variety of distinct lines. And we chose lines that were very different intentionally, just to get an idea of how broad a and you can see this is HER2 positive, two positive breast cancer cells. MCF7 cells, which are also a breast cancer line. And even in lines like HeLAs, HCCs are a colon cancer line, H460 are a lung cancer line. In all of those cases, if you treat with NEC1 to block the RIP1 kinase activity, you get a bump in the viability, and that bump only happens when cells are grown attached. So this appears to be relevant, not just in our MCF10A model, but also in our cancer. And I'm not going to show you these just in the interest of time, but we actually validated this with knockdown approaches. So if the concern was potentially that the inhibitor had effects, seeing data that were as a result of NEC1 impacting some other pathway, we see exactly the same thing if we do RIP1 shRNA, and we see again a benefit to the viability of matrix detached cells while having no impact on matrix attached cells. Okay, so this seems like our hypothesis that potentially programmed necrosis is induced in matrix detached cells, that our hypothesis might be right, that potentially matrix detachment causes the activation of RIP1, which would lead to a cascade leading to programmed necrosis. But we started to think about things a little bit differently and when we started to get these data that I'm going to show you now. So RIP1 typically in necroptosis will activate and phosphorylate RIP3, which can then signal downstream to a protein called MLKL. And that protein MLKL has the capacity to disrupt the integrity of the plasma membrane. So those are sort of the key, they're not the only players, but they're thought to be some of the really key players in necroptosis. So we wanted to look at RIP3, we wanted to look at MLKL, and if you're looking here at this gel, this is again in our MCF10A cells expressing BCL2. In this case, detachment does not change the activation or change the abundance of RIP3, which is interesting, very different than RIP1, but doesn't mean that that protein's activity is not still important. And you can actually do experiments where we antagonize RIP3 with an inhibitor, GSK872. We know that this inhibitor is working because we can measure the phosphorylation downstream of MLKL. You can see this here. But what's interesting is if you look at the viability, that GSK treatment has no impact on whether these cells live or die. So it has no impact in attached cells, it has no impact in detached cells. And what that tells you is that this is something very different than RIP1, that if we inhibit RIP1, then you see this fundamental difference in the viability of matrix detached cells, but RIP3 is completely dispensable. You can inhibit its activity and really don't have any impact on whether those cells live or die. And just to be clear, we did, again, I'm not going to go through all of the experiments we did here, but we did inhibitors of MLKL. You can see this compound NSA that I'm showing you here on the right. We did genetic knockdowns of RIP3, of MLKL. And in all those cases, RIP3 and MLKL are completely dispensable. You can inhibit the activity of those proteins, inhibit the abundance of those proteins, and you do not impact the ability of those cells to live or die when they're grown in detachment. So this leaves us with a model whereby potentially we have a RIP kinase 1 dependent effect, but a RIP3 or MLKL independent effect. So that got us thinking, what else could be downstream of RIP1? And there's a protein called PGAM5. This is a phosphatase known to be localized to the mitochondria that has been linked through previous studies to RIP1 biology. And so we started to think that PGAM5 might be an interesting protein for us to look at. So we did similar experiments where we looked at the abundance of PGAM5. What we don't see is we see no evidence that PGAM5 abundance changes in detachment. But interestingly, if we do knockdowns, if we do loss of function, what you're seeing here is shRNA against PGAM5. These are two different hairpins, the A and the B hairpin. We again see this significant change in the viability of these cells. So this is done in our MCF10A cells. You knock down PGAM5, and you see this really fundamental difference, again, only in detachment, where now these cells are surviving more efficiently. So this is very similar. This phenocopies what we see when we inhibit RIP1. So suggesting that potentially they're in the same pathway. So knockdown of PGAM5 recapitulates what happens when you do RIP1 inhibition or RIP1 knockdown, but how do we really know that PGAM5 is downstream? So in order to ask this question, we took we generated cells that were deficient in PGAM5. You can see this here. These are H460 lung cancer cells. We do shRNA to get a deficient PGAM5 deficient cell line. And then we mimic detachment by overexpressing RIP1. So just explain this again. Remember that detachment itself is a signal for the induction of RIP1. And that when you overexpress RIP1, when we do this in attached cells, that mimics what happens when you grow cells in detachment. So you can see here on the left, H460 cells, wild type or vector or PLKO vector expressing H460 cells, overexpressing RIP1, much like you would see during detachment, lowers viability. That's exactly what we would have predicted. But if you do that same experiment in the context where you have now removed PGAM5, what you see is the ability of RIP1 to lower viability is now lost. And what this tells us is the way we interpret these data is that RIP1, the ability of RIP1 to limit the viability of cells requires PGAM5 in some capacity. And so we expanded these studies again into a variety of other lines. And we see, again, much like we saw with RIP1, if we do loss of function assays with PGAM5, we see exactly the same thing in all of these different cell lines, where basically in detached cells, loss of PGAM5 gives you an increased viability during detachment. But what is actually going on? We're really, my background is really cell biology. We're interested in understanding fundamentally what's going on downstream of PGAM5. How is it that RIP1 signaling through PGAM5 is ultimately causing these cells to die? And so Mark got really interested in understanding if potentially PGAM5 itself could be altering the mitochondria. And we were sort of inspired by this idea that PGAM5 had been previously linked to the regulation of mitophagy, which is the intracellular turnover of mitochondria that's regulated by PINK1 kinase. So Mark wanted to look at is there a relationship between RIP1 and PGAM5? And he first looked at PINK1. Here you're looking at PINK1 is the kinase that's involved in the induction of mitophagy. Again, you're looking at attached versus detached cells. You can see a significant increase in PINK1 during detachment, which is interesting and consistent with the idea that we might be somehow inducing a mitophagy-like response. Interestingly, if you look at that PINK1 and then you treat with NEC1, so remember, NEC1 is a kinase inhibitor for RIP1. So in this condition, you see increased viability of matrix detached cells. The other thing you see is you start to see a cleavage product of the PINK1 protein, this lower molecular weight version. And what that tells you is that potentially, if you shut RIP kinase signaling off during detachment, that potentially you're starting to get some level of proteolytic cleavage of PINK1, and that proteolytic cleavage might compromise PINK1's activity. So Mark wanted to make sure that was in fact the case, we looked at a downstream substrate for PINK1, phosphoubiquitin. So you can see phosphoubiquitin levels are up at very high levels during detachment in the control in the MCF10A cells expressing BCL2 treated with the MSO. But when you bring in that RIP kinase inhibitor, you significantly compromise the activity of PINK1 as demonstrated by this diminished intensity of this band. So this leaves us with this idea that you turn on RIP1 and you somehow potentially will stabilize the PINK1 protein, but as soon as you inhibit the kinase activity of RIP1, the activity of PINK1 is now compromised, which means that mitophagy could be compromised as well, and I'll get to that in a second. Now, we did similar things. This was done in MCF10As. We did similar experiments in H460 cells. We see basically exactly the same thing. And again, if you look at the mechanism for how this happens, Mark really did some really very nice immunoprecipitation experiments to sort of figure this out. And what he determined was that as a consequence of the kinase activity of RIP1, you get a complex that forms between RIP1, PGAM5, and PINK1. And that complex protects PINK1 from being cleaved by a protease known as PARL. So if you disrupt RIP1 with something like NEC1 treatment, then that complex falls apart. PINK1 is now susceptible to proteolytic cleavage by PARL, which then compromises its activity. And what the hypothesis would then be is that ultimately, when you regulate the stability of PINK1 like this, that when you inhibit RIP1, that gives cells a benefit, they start to survive better. This may be due to the fact that you're inhibiting sort of a mitophagy response by compromising the integrity of the PINK1 protein. Okay, but what I haven't shown you is really, so all of that suggests that RIP1 is ultimately giving you that RIP1 is leading to a cascade that induces mitophagy. And that ultimately mitophagy may somehow be compromising the viability of cells. But what I haven't shown you is any assays that actually measure mitophagy. And again, mitophagy is the intracellular turnover of the mitochondria in autophagosomes. So it's analogous to autophagy. It's just that the cargo in the autophagosomes is the mitochondria. So what Mark did is he looked for evidence of mitophagy first by measuring the abundance of different mitochondrial proteins. So this is looking at TOM20. This is an outer mitochondrial membrane protein localized to the mitochondria. You can see that that protein detachment is a signal for that protein to go down. Again, that's suggestive or indicative of a mitophagy response, is that you would be turning over your mitochondria. When you inhibit RIP kinase with the NEC1 inhibitor, you block this, and now TOM20 levels remain high. So that's pretty decent evidence that seems to suggest that there is a mitophagy response during detachment, and it's connected to RIP kinase signaling. Here it is with shRNA. So again, here is our controls on the left. You can see detachment causes the loss of TOM20. If you either do shRNA against RIP1 or shRNA against PGAM5, you abrogate this, and now TOM20 levels are maintained during detachment. So this seems to, again, suggest a link between RIP1, PGAM5, and the induction of mitophagy. Mark also did this with MTCO2. This is a mitochondrially encoded protein. Again, detachment causes this protein to go down significantly, and you inhibit RIP kinase, you can see a rescue here where that protein now maintains a higher level. So all of this seems to suggest that detachment is a signal for turning over or for a diminished mitochondrial abundance, and that if you inhibit RIP1, you can rescue this. So there's a direct link between RIP1 signaling and mitophagy, or the mitochondrial abundance. So then what Mark did is he looked at the number of mitochondria by mitotracker. And you can see here, if you start on the left, mitotracker is just a stain for the number of the mitochondria. If you look in attached cells, you can see a lot of red. You go into detachment and that red disappears, again, indicating that your mitochondria you're diminishing the mitochondrial number. You do the same experiment where you've knocked out or knocked down RIP1 or PGAM5, and you can see that detachment does not give you that same loss of the mitochondrial signal. And here's the quantitation on this. So again, what that's telling you is the number of the mitochondria goes down significantly as a consequence of matrix detachment, and that if you inhibit either RIP1 or PGAM5, you can rescue this. So it again directly links this pathway RIP1 PGAM5 to the induction of some type of mitochondrial turnover. Now, do we really know that this is mitophagy? I mean, again, I've showed you that PINK1, which is widely known to induce mitophagy, is regulated by RIP1. I've shown you that there's a change in the mitochondrial number, but that doesn't directly tell you for sure that it is the mitophagy, it is a mitophagy response that you're ultimately seeing. And in order to address this, we actually went in and manipulated the machinery involved in the execution of mitophagy. So again, let me show you here. These are MCF10A cells in control. You can see that mitotracker staining, that mitotracker and attachment goes down with the attachment, and you block that when you inhibit RIP1 with the necrostatin 1 compound. And then we do this in cells that have been engineered to be deficient in ATG5. ATG5 is critical for the formation of autophagosomes. So this is basically a CRISPR-Cas generated line where now they're deficient in their capacity to undergo really any type of autophagy, but certainly mitophagy, where you could incorporate mitochondria into autophagosomes. And you can see now that you not only don't have that loss in the mitochondrial number during detachment, but the treatment with NEC1 completely doesn't matter anymore, that there is no impact of treating with NEC1 if you don't have intact autophagy machinery. You engineer these cells where you add back in ATG5, and now you've restored this. Your mitotracker signal goes down during detachment, but you treat with NEC1 and you rescue this. So what this tells you is that this is a bona fide mitophagy response. Mitochondria numbers are diminishing during detachment. They are rescued when you inhibit RIP1, and if you don't have the mitophagy or the autophagy machinery, then the ability of RIP1 to regulate viability is completely lost. So we thought this was a really nice way to sort of link these changes we're seeing during detachment to the regulation of both RIP1 and to the mitophagy machinery. Now, why is it that RIP1 mediated mitophagy causes cells to die? This is something that's really sort of interesting to us, and admittedly, we still don't fully understand exactly how this works. But why is it that if you induce cells in detachment to undergo mitophagy as a consequence of RIP1 signaling, why is it that these cells ultimately die? And we were interested in the possibility that the induction of mitophagy may impact the levels of ROS generating from the mitochondria. So we got these really interesting data. So we're using a stain called mitosox here, which stains reactive oxygen species specifically coming from the mitochondria. You can see that in detachment, that you see this significant induction in mitosox. So you see high levels of mitochondrial ROS. If you inhibit RIP1 or PGAM5, this induction of ROS is completely lost. And this is sort of perplexing because this sort of suggests that mitophagy itself is causing ROS, and mitophagy is typically thought of as a cellular response to eliminate defective mitochondria that are themselves causing our high levels of ROS. But we're arguing, and these data suggest, that the mere act of inducing mitophagy is enough to give you sort of an induction in ROS. And again, we don't fully understand exactly why this is the case yet. Again, we went back to these experiments where we looked at the machinery, the mitophagy or autophagy machinery, to prove that, in fact, it was a consequence of mitophagy induction that we were getting mitochondrial ROS. Here is our control cells. You can see the induction in mitosox staining with the MSO treatment. You can see that it's blocked with NEC1, which is, again, that RIP1 kinase inhibitor. But if you get rid of ATG5, which again is a key protein involved in allowing for the autophagy process to occur, detachment no longer gives you an increase in mitosox staining, and NEC1 has no impact. So again, that's telling you there's a direct link between the induction of mitophagy and the generation of ROS. Now again, you do your rescue experiment, you add back in ATG5, and you restore this, and now detachment gives you an increase in mitosox that NEC1 can regulate. So again, we do not claim to have fully figured this out yet. We don't understand exactly what it is about mitophagy that leads to high levels of ROS, but it seems like there is a fundamental link between detachment, RIP1 activation, the induction of mitophagy, and ultimately an accumulation of high levels of ROS. And that's something we're interested and continue to further pursue. So let me end with one last piece of data here. So all of this, if you take all of this cell biology that we've talked about how RIP kinase activation during detachment stabilizes PINK1, leads to the induction of mitophagy, and ultimately cells in detachment will die as a consequence of this. So you might predict then that if you disable this pathway in cancer cells, that this would enable them to more efficiently form tumors. And so Mark wanted to test this directly in a xenograft assay. And basically what he did is he generated lung cancer cells, H460s, that were either defective in or deficient in RIP1 or deficient in PGAM5, and then injected these into the tail vein of an immunocompromised mouse. And we chose this assay because we wanted to get an idea of how what happens when you put cells immediately into a context where they are going to be forced to survive during detachment. You're putting them directly into the circulation. Here you can see on the left, this is healthy lung volume. So as tumors accumulate in the lungs, this signal would go down. You can see the blue line shows you no change over time. That's expected. If you inject H460 cells, you do get tumors forming, and you can see this over time. You start to as you accumulate more tumor, the healthy lung volume goes down. But if you do this in the context of RIP1 knockdown or PGAM5 knockdown, that this is really accelerated and you get more tumor burden is what we would predict. If you actually do ex vivo analysis of the lungs, you can see here at the bottom, this is lungs from cells that were from mice that received just the H460 injection. Again, there is tumor forming, there is tumor on these lungs, but ultimately the abundance of that tumor, the amount of that tumor burden is significantly lower than when you either have the knockdown of RIP1 or PGAM5. And so what that would suggest to us is that this pathway is we've been studying may impact the viability of cancer cells during tumor progression, and that antagonizing that pathway might be a strategy that tumor cells use in order to survive more efficiently. And we do have evidence that this is in fact the case in patients. This is a study of lung cancer patients where you're looking at overall survival, and you're ranking, you're binning into tumors that are patients that either had low or high expression of RIP1 in their tumors, and you can see the prognosis or the overall survival of patients that have low levels of RIP1, which would thereby enable those cells to potentially survive a little bit more efficiently, that their overall survival is actually much lower than patients who have high levels of RIP1. Now, we're not necessarily suggesting that that's what happens in the majority of cancer patients. You could imagine a variety of different ways or strategies that cancer cells would use to turn this pathway off, but we do think this is at least evidence that potentially there is a clinical benefit to maintaining this pathway in patients, and potentially thereby allowing cells to be killed during tumor progression rather than them to survive. Obviously, this is entirely correlative, but we think this is really interesting and in line with our hypothesis on the basis of our xenograft data. So I'll end here just with a quick summary. Just a brief, I didn't go through all of the pieces of the mechanism that Mark figured out. There's some of those that's been filled in here. This paper was actually just published a couple months ago, so if anybody's interested, obviously take a look at that manuscript. But in matrix-detached cells, you have an activation of RIP1, you have the stabilization of PINK1, which leads to the induction of mitophagy. There's an increase in mitochondrial ROS, which leads to cells dying. And then if you inhibit this pathway, you allow for PINK1 to be proteolytically inactivated. This basically allows for an inhibition of mitophagy. And one of the things we do know is this restores some of the reducing potential in the mitochondria, particularly from IDH2 to produce NADPH, and we get some sort of protection from mitochondrial ROS. We know that this matters in vivo because you inhibit this pathway and you can enhance tumor formation in vivo. And, we're really, again, we obviously don't have everything figured out in this case. There's still a lot of really interesting questions that we're pursuing, But we think this is a really exciting finding that tells us a little bit more about how it is that cells survive during matrix attached conditions, and ultimately what that means, that may have consequences for how cells progress, cancer cells survive during tumor progression. Let me just acknowledge a couple people, and I'm happy to take questions if anybody has any. I mentioned this is really just sort of I've got a lot of people from my lab on here. So again, I mentioned this before. Mark Hawk is the grad student who led up this study. There were other people involved, but Mark really was the primary driver of this really nice. I have 3 other grad students right now, Matthias, Jordan, and Lisa, all really phenomenal. Have a great technician, Gemma. I have 5 undergraduate students working with me, and that changes depending on the time of year. We've had some great collaborators on this. Ralph DiBerdinis and Lei Jiang, previously from his lab at UT Southwestern, were really great collaborators on this project. Mike Overholzer at Sloan Kettering, and Jens and Monica from his lab. Matt Lee and Sarah Chapman helped us here at Notre with all the in vivo studies. Got a variety. Again, these are some former lab members. Cassie Gorsuch also worked on this project significantly, and again, a variety of. I've really been very lucky to have such amazing grads. These are the 5 that I've had so far. These are some previous undergrads that I had. Our sources of funding, and again, I'm happy to take any questions that
Thanks, Dr. Schafer. It's a pretty nice presentation. Yeah.
Thank you. I appreciate it.
I have one question. Like during metastasis, right? Like cells leave the tumor and go to the other organs.
Right.
So when you say like cells are released, cells get died when they're coming out from this tumor.
Right.
So have you looked into that aspect? Like what does it different in there compared to what you are seeing in here?
I think they're probably ultimately related. I think that there is no question that cancer cells, when they leave primary tumor and when they start to travel in the circulation site, or even when they see the second site, that these death pathways are going to be important. And what I mean by that is turning those pathways off. So we think that through our studies, as well as a variety of other groups who are really active in the area, that potentially, if you can find a way to sort of interfere with their capacity to shut these death pathways off, that might be an effective novel therapeutic strategy. And it could happen either by causing those cells to ultimately turn on the anoikis pathway, or it could happen by rewiring their metabolism in a way that impacts their ability to induce this mitophagy response. So we do think those things are related because those environments that are counter depression are that's a stressful condition where shutting these death pathways off is going to be critically important.
I think I don't have. It's a very nice presentation. I learned a lot from your presentation today. I really appreciate you taking time and talk to us about this your work. And so, do you have any can I give your email out so that if at all people are interested in talking to you, I can do that?
Please,
okay.
Of course. I really appreciate the opportunity to participate here today, and congrats to you on putting together such a great community and really making it easier for scientists to communicate. I think this is a really great idea, and I'm just happy to be part of it.
So thank you so much. So it will be on our website as soon as we are done, and you can go and watch it, or you can let others know about it. And good luck with your research. Thank you so much.
Thank you. I appreciate it. Take care.